In this video, we will discuss about the glycosylation and its relationship with pathogenesis. So briefly, we will discuss how this glycosylation pathway helps in the viral pathogenesis and what are the strategies host cell used to counter those uh, viral pathogenesis. So it starts with the endoplasmic reticulum where the glycosylation or mainly end-linked glycosylation begins. After that, it will move into the Golgi for further post-translation modification, or in other terms we can say uh, the initiation of O-glycosylation and maturation of the N-glycosylation that happens in the Golgi. And after Golgi, this viral glycoprotein will form. So one things that it can do is viral will release, for example, the influenza will release into the cellular matrix, and one of the strategy that used to evade immune is by secreted themselves into small glycoprotein parts, like it is shown here. This is uh, one of the glycoprotein that are found on the surface of the virus, or are found on the envelope of the virus. So if this itself glycoproteins is available, then immune system might not be able to recognize whether this is the part of the virus or this belongs to the body. The immune system will not be able to differentiate, and this strategy has been used by the virus such as HIV, DENV, EBOV, uh, and other strategy by that used that other strategies by which glycosylation help in the viral pathogenesis uh, share, is by shielding glycoproteins with the help of glycan, so that uh, the immune system of the host cells will not be able to interact with the antigen of the glycoproteins of the virus. And some of the heavy glycan shieldings are being observed in the HIV, arenavirus, Ebo, coronavirus, influenza virus. In these viruses are heavily glycosylated, so that it's difficult for the immune system to recognize the glycoproteins. It's difficult for the immune system to uh, pass this glycan shielding and reach to the glycoprotein, so that they can generate an immune response against those glycoprotein antigen. So this is one of the examples that uh, immune response or antibodies will not be able to interact with the virus glycoproteins, and they will be shedded by the glycans, and these glycan structures are similar to the glycan structure found in the host cells. And other strategy is by with the help of this attachment factor and enhance uptake by host cells. So in this case, what happens is that these host immune cells will this viral glycoprotein will recognize the carbohydrate structures that are present on the host cells, and they will replicate or mimic the carbohydrate structure similar to the host cells, so that they can enter the host immune cells and weaken the immune cells. So this is strategy is known as attachment factor and enhance uptake by host cells, and this strategy has been used by alpha virus and Fabo virus, where they weaken the immune system by entering into the immune system and by entering into the immune cells and making them dysfunctional or less functional, or in other terms inefficient. And other things that they do is that this is similar to this attachment factor and enhancement uptake by host cells. However, in this case, what happens is that these viruses structures will target instead of the immune cells, they will target the other host cells other host cells present within the that are present within the host, so that they can infect other cells. And the strategy that they use is the same strategy, that is they mimic the glycan structure similar to the glycan structure that are present in the host, since they are using the same machinery of the host cells. And these glycan structures will help them interacting with the receptors on the host cells, and that helps in entering endocytosis or entering of the virus within the host cells in the cytoplasm. Next one is the soluble lectins of innate immune system and complement activation. So this is some of the this are the strategies that are used by the immune system to interact with this kind of glycan shielding, or we can say viral pathogenesis prevented by glycosylation. That is, they use soluble lectins uh, so that they will be able to interact with the glycoprotein antigen and generate immune response against it. As soluble lectins can reach to a more condensed state compared to the uh, transmembrane compared to the membrane lectins. And other things that they use is other strategies that the immune system use to counter the immune system use immune system can use to counter this kind of heavily glycosylated virus is the glycans, is to generate antibodies against the glycan epitopes that are specific to the virus. So in this case, what happens is that whenever the glycoprotein virus is produced, though most of the glycan structures are similar to the host cells, there are some glycan epitopes that are unique to that virus. So those unique epitopes can be used to generate antibodies against those epitopes, and that can help to determine the virus. It can help immune system to determine the virus and can act against it. And these unique epitopes has been found so far has been found in HIV, DE, and V viruses. This strategy also can be used to counter these kind of viruses. And these two strategies can be used, and the other strategies and the other techniques are shown how this glycosylation helps in the in evading the viral pathogenesis. So I hope you have understood or got an overview about how this glycosylation and pathogenesis are related. And if you need to understand these things in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.